Welcome, everyone. Pleasure to have you here live, or if you're watching this recorded, thank you very much for joining us. I'm going to be one of your two hosts today, along with Kathy at the University of Calgary. I'm currently here at the University of Sydney. And I want to, before beginning, pay my respects to the Gadigal people of the Eora Nation, which is where I'm currently work and live. I want to pay my respects to elders past, present, and those who will emerge. So today we have Dr. Daniel Strech providing a presentation looking at translational research ethics. So very excited to have Daniel here. And then we'll have an intervention by Dr. Chris Doig, who's at the University of Calgary. He'll be sort of taking us through more of the actual, his experience with some of the ethical issues around translational research ethics. So next slide. So for those of you who are joining us for the first time, we're a multidisciplinary research platform funded by CIHR to look at various facets of microbiome research. We have several different platforms, as you can see there. The role of this project and the role of this group is to support microbiome research in Canada and internationally across those platforms, and to be sort of a place where we can sort of think through some of the complexities of these issues. And there are the various leads for the platforms themselves. Quick promotional note, we're having a research meeting conference that everyone can attend. That's June 2nd to 3rd, having IMPACTT, Advancing Microbiome Research. It sounds like an action movie. It's like the revenge of microbiomes. That should be like part of it. It'll be in Calgary, Alberta. So looking forward to that. So, yep, there's Kathy and I. We're going to be your webinar host. Kathy's going to be running the Q&A as well. And I guess now is a good time to introduce our speakers. So our first speaker is Dr. Daniel Strech, who studied medicine and philosophy, worked as a physician for a few years in Germany. And then he started as a researcher in bioethics and meta research. From 2008 to 2018, he worked as an assistant later as a full professor at the Hanover Medical School in Germany, where I was lucky enough to work with Daniel for a bit. In 2018, or since 2018, Dr. Strech is full professor of translational bioethics at the Berlin Institute for Health at Charité at the University of Berlin in Germany. And he's also deputy director of the Quest Center. And his research teaching and consulting activities focus on ethical and regulatory issues in medical research and public health and health policy. So we're very happy to have Daniel here with us. We also have Dr. Christopher Doig, Chip, who's a professor at the Cumming School of Medicine at the University of Calgary, received his MD with distinction from the University of Saskatchewan, and he currently specializes in internal medicine and critical care medicine. He's acted as department head for community health sciences in the Department of Critical Care Medicine at the University of Calgary, recipient of numerous local, provincial, and national and international awards. And he has extensive research and publications covering a breadth of topics, including translational medicine, clinical trials, and medical ethics. Both of our speakers are highly distinguished, and I will let you to look up their bios later on. But for now, I'm going to turn to Daniel. Daniel will speak for a little bit, and then we're going to have a brief intervention by Dr. Doig, and then we'll have time for questions and answers. Daniel, over to you.
Yeah, thank you very much for this very kind introduction, for the invitation, of course. It's a pleasure for me to be here. While speaking, I try to share my screen. Do you see my screen now? I see some thumbs up. Okay. Yeah. In a brief discussion with Diego, I came up with the idea and the suggestion to, I try to do two things here because it's an ethics seminar with people who work in translational research. I will try to do two things. More briefly, at the beginning, introduce so-called translational approach to questions within bioethics. I will explain what stands behind this term, but then I will pick one example, as I would argue, very prominent example or issue of the ethics of translational research, and where we did a lot of translational research on this issue and describe this in more detail. So I start with this introduction about, oops, can I forward here? Yeah, this introduction about what I mean when I say or claim that there is an often neglected approach to bioethics that is this more translational approach. So let me briefly outline this, and the intent maybe is also that you become more aware about how often very important bioethics work that of course relate to your work is could still be further developed into something that becomes then maybe even more practical and practice oriented. So this is a little bit the goal here, to raise some awareness for this. But starting here, bioethics, when it comes up to biomedical research in general, could be introduced like this. You would say, what is biomedical research and what is your goal? Then people would explain, primarily it is about generating value, right? There are a lot of ways on how biomedical research in all its different domains aims for knowledge gain, and this knowledge gain finally should result into a value for science via this knowledge gain for better disease understanding, better understanding about therapeutic options, and ultimately, at the end of the day, also as a value for society when we realize to also improve healthcare for patients. And then you would say, yeah, sure, this sounds very, very desired, and it would be unethical not to do this, not to try to generate this value. Is there anything else? And people would say, yeah, for generating this value, we have to deal with a lot of sensitive topics. We need to do research with animals. We often, increasingly now, do our research based on maybe big data sets on clinical data, patient data, and we also do research with human subjects in clinical studies. And for this reason, we talk about the ethics of translational and biomedical research. We would need to balance this in a certain way so that we have the possibility to generate this value, but at the same time safeguard these participants, so to say, and subjects that we need for our research. And therefore we developed ethical principles. You are well aware of them when you work in animal research. You are aware of this ethics framework, the 3R framework: replacement, reduction, refinement. Big data commonly deals a lot with broad consent issues, how to use and access data. In clinical research, since decades, we have principles such as informed consent, and this RBA here stands for risk-benefit assessment, which will be the main topic in the second part of this talk. And sure, for all these fields, we have several transparency guidelines. And there is more, of course. I just mentioned this here because this is, of course, builds part of maybe the most fundamental approach towards bioethics, that we develop principles, ethical recommendations, theories that should be applied in practice to make the practice better. And nowadays, maybe these very broadly defined principles here that I listed, they, of course, they are not reinvented every day, but nevertheless, we do a lot of conceptual normative work in bioethics to specify these principles, because biomedical research with animals, with data and clinical trials change, and we need to adapt and specify these principles. But it's all about conceptual normative work that is ongoing and very important. you have a completely different approach, a second complementary, not competitive, a complementary approach that is more empirically oriented, where you do interview research, survey research with all the different stakeholder groups, and ask them about their attitudes towards these potentially relevant principles, right? You can ask people about what you think is broad consent, a good concept to deal with biobank research and so on, what you think an informed consent template should entail. And there is a lot of important research going on. So this is all not yet translational bioethics. I will come to this in the next part. Just want to give some examples that in all these fields, we see a lot of current research here. A concepts paper, a purely conceptual normative analysis on one current topic, that is, do we need upper limits of pain and suffering in animal research? And then here, these two authors do a normative analysis of this. And an important, very timely, purely conceptual work within bioethics. Here's another example from my work, where I had to deal with the question, do we really need ethics committees reviewing each individual biobank material access inquiry? Because if a research ethics committee, an IRB, already reviewed the consent materials and the data protection model and approved the whole biobank, why do we then need to include a research ethics committee for each individual biobank study with the material? And I argue a little bit against using IRBs for each individual release of material. But this is a purely conceptual normative piece. And then for attitudes research, there are even reviews of surveys and interview studies that asked the different stakeholder groups about their perspectives on what is ethically relevant in biobank research and so on. But this is attitudes research. Or together with Diego, we did this study where we interviewed stakeholders in animal research about pros and cons, strengths and weaknesses for having also at least some parts of animal studies pre-registered. So it was an empirical analysis to better understand what could become relevant for the ethical analysis. But all of this, both the conceptual work and the attitudes work, finally serves the goal that we come to good justified recommendations on what we should do in the practice. And I hope that you already now have a picture about what the translational approach here might mean. At the end of the day, we have to go from, not here from bench to bedside, but from the theory to the practice, from the desktop where we develop this into the concrete decision making. And this needs to be applied in the practice. We have also regulation going on that's trying to implement these principles into practice. And this also comes with time and costs. So it's something that takes a lot of time to deal with all these principles. And therefore, there are these very obvious two questions. The way we currently implement these principles into practice, is this effective? Is it working? Do we really do something that is meaningful with regard to what we planned or what we wished to do as consent, risk-benefit assessment, and so on? And to ask also, is it efficient? Do we maybe implement it in a way that is just too time-intensive, too costly, without really value and effect for this implementation? So this is this part of evaluation and implementation research, so to say, that I would frame as a translational approach to bioethics. And it's not competitive here, right? It's not that one is better than the others. We need all these three approaches to bioethics and combine it to make this field professional and practice-oriented. So this is my idea. If you look into the literature of bioethics, we are still working on a review of how often you find these more evaluation and improvement studies. I would argue, I don't have data yet, but it's a very little field. You don't have a lot of studies that explicitly try to understand how certain principles are implemented. But I need to get more evidence on this. So, sure. I also need to give 2 examples here. So one example, which is not from my group, is, but I find nicely fitting here, is from a group from Duke University. So they took the already accepted principle of consent for biobank research, but they studied how the consent currently is going on. They identified several challenges for the implementation of this consent form. It was not so effective, but also not very efficient. And they tried to simplify the consent form to finally reach both goals, to make this more effective, that those that donate biomaterial better understand what they are consenting on, but make it also more efficient because it was simplified, a little bit shortened. So this was I would say, very ideal translational bioethics study. And the study that I will use as the example for my second part of my presentation is this one, where we, together with colleagues from McGill University, had a look into investigator brochures for phase 1, phase 2 trials from different therapeutic areas. And our aim was to understand how is the data and the evidence presented that we need for risk-benefit assessment prior to phase 1, phase 2 trials. How is this currently implemented? How do we do a risk-benefit assessment? Or here, more specifically, on what evidence basis do independent ethics board and regulatory authorities do risk-benefit assessment? And we found a rather concerning situation, and I will go into this in my second part. So yeah, and the second part more or less starts now. So this was this brief introduction to translational bioethics. The second part is divided in these 3 parts. I will start a little bit with background information. Why is reviewing preclinical research prior to conducting clinical trials so important from an ethical perspective, to make clinical research ethical, and what are the past developments that pronounce this very specifically, and why investigator brochures in the current shape, in the current fashion, do not really allow something that we might label a meaningful review. And I then come up with a very broad proposal, but I think there much more work is needed in this area. So I start with this more background thing. I thought I do it in a more systematic way again, so that we have common ground when we are discussing what I introduce later. So what makes especially early clinical research, phase 1, phase 2, ethical? It is the already mentioned value, right? We want to generate knowledge gains. We want to generate value, maybe even for patients. So this is something that we want to have, but of course you have to justify this a little bit to yourself as a PI who is launching a clinical trial, but you also want to engage in a risk-benefit assessment, because the value, the potential benefit that you want to generate for patients and knowledge gain also comes with risks for the study participants, and therefore we have this risk-benefit assessment. So both are very established principles in research ethics: social value and the balancing with potential risks. And since the 1970s, 80s, via the revisions of the Declaration of Helsinki at this time, it became a principle that not only the PIs who are conducting the clinical trial do this risk-benefit assessment for themselves. They, of course, have to do the risk-benefit assessment themselves, but we should include independent reviews, such as ethics committees, but also regulatory authorities do this independent assessment on whether this trial is sufficiently justified to be conducted. We have other types of reviews who engage partly more, partly less in this risk-benefit assessment. There are, of course, other principles such as informed consent and fair participant selection, but they are not in the focus of my talk here. So these 3 principles are relevant for me. And yeah, so to do risk-benefit assessment, we need some sort of evidence we can base the risk-benefit assessment on. Otherwise, it would be purely speculation to say, I want to do something good. I have an idea, here's a treatment option, and then we say, okay, we find this is a good idea, but we will say, no, can you please show me some evidence, probably from preclinical research, that it's sufficiently safe and there is sufficient clinical promise, at least in this. And this mainly stems from preclinical research when we talk about very early clinical studies. So far, the background for how preclinical research ties together with this several ethical principles that come into play when thinking about, shall we start this study? Now, we know from a lot of research on research and also regulatory research that we have, of course, failures in clinical research. There should be failures. If there are no failures, then we would say we should probably not do research anymore. If everything works, then we could directly go to healthcare. So it's sure we need to have failures in clinical research, but a lot of people complain that we have failure rates at a very high level. There might be something that does not work well enough. And this very prominent study here, one of the biggest studies that followed up the drug development of several thousand drugs, have these data here that are also the most prominent data, and often people refer to these data, saying that we are relatively successful with clinical research when we are testing the safety of a drug. So when we go into phase 1 clinical research, you see a 64% success rate, meaning 60 or 2/3 of all studies on phase 1 level make it to phase 2, because when we tested safety in phase 2, it seems to work. And people, of course, then can conclude our preclinical research that informed the safety considerations seem to be a relatively good job. They seem to predict what is going on in clinical research, not perfectly, but on a high success level. Better, it's good that we do these safety studies preclinically. They inform it. But the big challenge comes at phase 2, when we for the first time assess the efficacy of this new drug. There we have this high failure rate. Only 1/3 makes it then to phase 3 because they fail because of a lack of efficacy, despite the fact that we did a lot of basic and preclinical research that demonstrated that there is a clinical promise, but then it fails in humans. When it goes to phase 3, another 40% fails, and this is also mostly because of the lack of efficacy. It's very rare that phase 3 clinical trials fail because of safety concerns. It's then mostly because of the lack of efficacy. So this is the background, and then discussions increased over the past decade to say, look, there are, of course, a lot of ties between preclinical and clinical. The way we conduct the preclinical efficacy research becomes relevant, of course, for decision-making on when to launch clinical trials. Here, the colleagues with which we did this IB analysis also published this more conceptual piece in Nature, describing why we should look more on the preclinical data that give us a clinical promise before we do phase 1, phase 2 trials. There is something that needs to be improved. While many people would say, what recommendation is this about? Isn't it something that we take for granted, that people, of course, consider drug efficacy before first in human trials? These authors argue, no, probably not. We are currently maybe not doing this, at least not in a professional, systematic, trustworthy way. So this is the point for why preclinical research becomes so relevant, and we have to deal with it. But there are two discussion areas, I would say, where, again, over the past decade, several concerns rise about the robustness and the usefulness of the data stemming from very preclinical animal studies. So if we broadly distinguish these concerns, I would say one area of concern is that we maybe do not know a lot of data that has been produced, but that is then finally not published, and is maybe also not reported in investigator brochures and study protocols, the so-called publication bias challenge or concern. Interestingly, there's not a lot of research out there on how many animal studies that are completed finally become published. This is a typical question in clinical meta research. There are, I don't know, maybe really hundreds of studies that investigated how often completed clinical studies finally become published. And of course, they found that many do not publish their results, and a lot of measures were invented, so to say, over the past decade, such as pre-registration and certain obligations for results reporting in the field of clinical research. But in animal research, we even still do not have the most basic information about what is the extent of a publication in this field. There are some surveys, such as this one, and I realized that I did not put the publication year for this paper here, sorry, but it's almost 10 years old, this paper, where a group in the Netherlands surveyed animal researchers in the Netherlands and asked how often do you think are animal experiments published, and those animal researchers themselves said we think about 50% of the animal experiments are published. So this is a survey study. We don't know how much it reflects the true number. This study took another approach, and again, I don't know why, but the publication here is not here, but this is also almost 10 years old. They used a more statistical approach to investigate this publication issue. They did a meta-analysis of a lot of animal stroke studies and showed via statistical measures that there is probably a publication bias, which goes into a major overstatement of efficacy. If you only include published studies, there is at least this concern about this publication bias. And a major overstatement of efficacy could, of course, explain a little bit why we have maybe such a high failure rate at the phase 2 level, because if we often think that the efficacy is much higher than it is, then we might maybe sometimes do studies, clinical studies, large clinical studies, in situations where the efficacy is less convincing. What we did over the past years is, I would say for first time, a very direct assessment of publication rates in animal research. So we really followed up completed animal studies from 2 universities in Germany to later publication. I could tell more about this because this was a very interesting process to make this study possible. A lot of conversation and also a lot of positive feedback, I have to say, from the animal research field in Germany that supported the study. And finally, we had 2 animal research facilities who worked together with us to make this possible. So we followed completed animal studies up for later publication and found that about 69% of completed animal studies published at least 1 results publication. We also did a check with the principal investigators to confirm this. So including all these measures, we came up with this number. Interestingly, we were not aware when we did this study, but I became aware shortly after we published it that more or less the same study was done in the Netherlands at Utrecht, and they found more or less a very similar pattern. 60% of the completed animal studies in the Netherlands published at least 1 results publication. As I said, it's a topic in itself, because when we finalized the study, we then realized that it's maybe questionable whether this publication rate number here reflects a valid measure for publication rate. This is because in Europe, in Germany, in the Netherlands, but also in many other European countries, an animal study, and this was our sampling, an animal study that is approved and then completed at a certain time includes a lot of animal experiments. So it's not like in clinical research where a clinical study has 1 experiment. An animal study comes with maybe 5, 6 experiments. So if we say we found for each animal study 1 results publication, we might heavily overestimate the publication rate at the experiment level. But it's very difficult for several reasons. We are currently investigating this, how to get a sample of animal experiments and not of this overarching study thing. I don't know how much this is relevant for Canada, or whether you have a completely different approach in this separation of studies as bigger packages of several experiments. Interestingly, this Dutch study here also looked not at the level of experiments and how many of them have been reported, but they looked how many of the animals that were approved in the overarching animal study then finally showed up in the publications. And they found that the publication rate for the animals is, in this case, was 26%. So the true publication rate in animal research might be in between 26 and 60%. If you go back to the survey of the animal researchers, these these animal researchers themselves thought that 50% of experiments is published. However, I just wanted to frame it's an interesting field that needs more investigation, and also some conceptual work about what does publication and full reporting in animal research mean. There are a lot of other interesting issues here. Okay, this is the publication type of concern, so to say, or challenge. The second part is this. How valid are the data? How robust and reproducible are the data? So the reproducibility crisis became a term in this regard. And it more or less reflects this idea that, of course, we have risk of bias, not only in clinical research, also in animal research, but we have several measures that we can use to reduce the risk of bias. Randomization, blinding of outcome assessment, sample size calculation, this is this SSC. And here's a study that investigated on a random sample of published animal studies how often you find randomization, blinding, sample size calculation described in the methods part. And you see that this was found not very often. Blinding and sample size calculation is very rarely reported. This improved a little bit over the years, up to 30% in the newer cohort. But you see that even this newest cohort here is almost now 10 years ago. So in other studies, however, you still see these patterns that about 30 to 40% of papers report randomization, blinding, and sample size calculation is often not mentioned. This group here more or less did the same thing, but looked at not at published papers, but at the approved animal experiment applications in Switzerland, and found more or less the same pattern. It's important to know this because people might argue, yeah, it's maybe not reported in the paper, but in the application, you will find randomization and blinding described, but they also did not find this described in the application. But again, the same cohorts, right? The newest cohort from 2012. Based on these findings, and also in parallel, there were this interesting thing that not only essayists or meta researchers that do research on research pointed to this challenge, but there were two very influential and highly cited papers from authors that were all representatives from important drug developers, BioHealthcare and Amgen, and both wrote in their papers, which are also now almost 10 years old. We try to reproduce a lot of landmark cancer studies published in Nature Science, and we were able to do so in only 10% of cases. 90% of the replication efforts failed, and we believe this impacts. This is part of why we have this high failure rates in clinical research, and we believe this is because of these two areas, that we have this publication bias probably going on, that a lot of negative, undesired results are not published, and if we see what is published, we then often find studies that did not use blinding and sample size calculation. So it's not only the ethics folk, it's also in this case the private funders who see this as a challenge. Yeah, NIH did a workshop on this and came up with recommendations that the minimum that studies, preclinical studies informing phase 1, phase 2 research should do is report on sample size estimation, randomization, and blinding. So there were these consensus meetings stressing this. I skipped this one. This is something that maybe does not fit too well with the narrative right now, and I want to speed up a little bit. So, but the remaining 2 points are quicker. We, based on this background, we then tried to get access to investigator brochures, right? You probably know what investigator brochures are. When you are planning a clinical study, clinical trial, you develop a study protocol, which is not too lengthy, but you also have this investigator brochure. It can have several hundreds of pages that describe all the supporting evidence, which is mostly for phase 1, phase 2 preclinical. And we were able to sample a little bit more than 100 investigator brochures that were approved by IRBs in Germany for phase 1, phase 2 studies, and to have a look into the sections that describe the preclinical efficacy studies that seem to be the supporting evidence for launching this phase 1, phase 2 trial, and is the evidence that the ethics committees and the authorities in Germany use to make decisions about whether there is a favorable risk-benefit assessment. So we went through these 100 investigator brochures and searched for preclinical efficacy studies only, right? We did not check for safety studies. We did this later and found more or less the same pattern, but this here was on preclinical efficacy studies. We found a little bit more than 700 of them. So 7 preclinical efficacy studies in average for one investigator brochure. And then the following core results. So one core result was that only about 11% of these 700 preclinical efficacy studies reference to a published report. So there was no published report available for these, for 90% of these studies. One could argue this is maybe not surprising because it's really commercially sensitive. It's still in development. And it's another question of whether this is legitimate, but it points to the fact that it's so important now for those who are reviewing these studies, the IRBs or the authorities, that they look into the quality of these studies. Nobody else did this, right? You could argue if all these studies were already published in peer-reviewed journal, you had at least some quality checks of whether these are trustworthy studies. But here, the vast majority was never sent to an independent review. So the IRB or the regulatory authority needs to understand what these studies are about. And this is why we found it important to check whether the investigator brochure says something about how these studies try to reduce risk of bias, but we found that they do not, more or less. We never saw something about sample size calculation, whether these studies used a blinded outcome assessment was never reported. 4% of them described that the study was randomized. And even the most basic information, for example, the sample size in these studies was only reported in about 1/4 of all of these preclinical studies. So in most cases, the IBs explain to the authorities and to the IRBs only that in a mouse study, we found that our drug works. And it comes without any information that you can use to critically appraise whether it's a trustworthy study. We also found that the vast majority reported positive outcomes. 90% of these 700 preclinical studies were described the desired outcome. 6% described a non-desired outcome, and 4% were unclear to rate here. Again, one might argue, yeah, look, this is not surprising because we are in development of a drug. So why should people start a clinical trial if the preclinical evidence was negative? It would be unethical to do so. Sure, you need positive preclinical efficacy studies, but the question is whether the 90% here reflects something that is meaningful. There are 2 concerns here. The first is statistical. The sample size, if reported, was about 7 animals per group. And if you have these low sample sizes, just by chance, you need to see some negative results. It's hard to believe that even if these were false negatives, there are no negative preclinical efficacy studies, but everything is from the beginning always positive. This is hard to believe, alone for statistical reasons. But then second, you need negative efficacy studies to understand the barriers and the boundaries of your drug. At what dosage, at what time becomes your drug non-effective? I mean, if you have no information about where the boundaries of your drug is dosage and so on, how do you do a good decision about whether this dosage that you use for this study is the right one? You need negative efficacy studies to better understand whether it's a well-chosen drug scheme, but it's not in the IBs. So yeah, summary. So I would say what we found points to the fact that investigator brochures in their current shape do not really allow a meaningful review of preclinical evidence. If there is preclinical evidence presented, it has low sample sizes and often lack negative studies, which is concerning. Why do oversight bodies such as FDA and the IRBs do not complain about? Why are they happy with the current situation? Hard to know. I mean, we did some informal discussions, of course. We can maybe come back to this in the discussion later. Often I hear back that people just do not review the supporting evidence for efficacy. This might explain why nobody complains. If nobody looks into the data, then of course they will also not complain that if they would wanted to use it, they would not be able to do this. But a lot of reasons for why this might be the case. We heard that they trust the sponsor, that they will not launch a clinical trial, which is very costly, if they did not make sure that there is a certain promise. The authorities, the ethics board need to check that it's safe, but whether it has a clinical promise is something that we need to believe the sponsors checked sufficiently. Whether this is justified, this hope is of question, of course. Then we also heard back that we lack the expertise and the resources to do so. So we have, of course, at the authorities, we have a lot of staff that looks at safety, and there we have standards and best practices and protocols. But for efficacy, it's so drug specific. You need so much drug specific expertise to make judgments about the efficacy studies. We are not able to do this at the oversight bodies. And of course, people make this point to say, look, yeah, safety studies are important, but efficacy studies, preclinical, we really question whether this is of any predictive value for the later clinical trial. And this is why we don't give too much attention to the efficacy studies. But if this is right, then why are we doing this at all? Why do we do animal efficacy studies if at the end of the day, nobody cares about the results and whether it's trustworthy or not? So a lot of question marks, I would say, about the current situation. And this is not a whole other part, but very briefly, a very broad recommendation is that we maybe need something such as a good justification practice. Right? I use this term because we have so many good practices. We have, and this is this typical picture that exists in different versions, just showing where we come from preclinical to clinical. You all know that we have a lot of good clinical practice guidelines that describe how to do a clinical trial. We have good laboratory practice guidelines that describe how to do a good laboratory study. But what is lacking in the field of translational research is something such as a good justification practice. How to make good decisions about, I am ready now, I am ready to translate. There is no guidance about how to make these judgments. If you look into textbooks for drug development, there is no chapter about how to decide when you are ready to translate. It's just about how to do preclinical, how to do clinical, and how to deal with regulatory things, but there is no guidance on how to make a good justification that now I'm ready to translate into human trials. So my last slide, I would say something such as a framework, conceptual work as well is needed in this area to support those who engage into reviewing the preclinical evidence. Of course, this makes things again more complex, and translational research is already so complex. But we are talking about one of the most fundamental tasks, I would say, in making clinical research ethical. If we have not made our homework here, then all these downstream things, such as risk-benefit ratio, informed consent, are heavily challenged. How can we do a good informed consent if we don't know whether the study is justified at all? So it's, I would say, nevertheless, worth to think about how to improve the situation. And therefore, we also not only have to improve the ways the evidence is presented in investigator brochures, we probably also have to improve the evidence itself, because when we look at the evidence, it's often with low sample sizes. We might at least fear a certain selection bias in what is presented there. And there are several measures for how we could deal with it. Maybe I skipped this. And we are currently doing further research, also stakeholder interview research, to better understand how to implement this improvement of risk-benefit assessment, because of course we are addressing a field that is already working since many, many years, and it's not easy to implement new structures in this field. But this is more or less one point of translational bioethics, and I hope I could make my points until here. Thank you very much for your attention.
Thanks very much, Daniel. That's terrific. unbelievably frightening in its own way. I think it's somebody put it down there. So I think you've given us a lot to think about. I'm going to now bring forward Dr. Doig. Please, yeah, thoughts, comments, what has been your experience?
You betcha. Thanks, Diego. So first, let me say that was a fantastic presentation by Daniel, and very thoughtful. So when I was approached and asked to provide a few comments to this, I discussed with those that asked me. Probably what I would do is take Daniel's presentation, just make some comments, reflections upon what he had presented. So first, let me just say, for the scientists, how much I respect all of you. Bluntly, I'm not smart enough to be a scientist, or I'm not obsessive compulsive enough or detail oriented enough. And that's despite in the ICU where I do most of my work being called a type A personality and having a reputation about being a bit obsessive compulsive or anal retentive. But one of the fundamental things that I respect about scientists is the curiosity-based research, the ability to come up with an idea and pursue an answer, starting off with an idea where you don't know the answer to where you finally establish an answer. And that reflects back on the value to science that Daniel said, the ability to contribute to scientific knowledge and understanding. But at least scientists privileged to work with have always told me they would love for their career or their work to do a little more. And I think that says the value to society. And although there's value to society for the contributions to science in and by itself, the implications to potentially find a treatment, a therapeutic intervention, something that helps a disease condition often is something that drives at least medical curiosity-based research. And so that's from the bedside. One of the beauties, of course, of the bench, despite the complexity and work that it takes, is that in some way very controlled environment, whereas the bedside and doing research at the bedside, and Dr. Nasser is on the Zoom talk. At the bedside, it's a very dirty, chaotic, uncontrolled environment. And so these findings to now translate them into clinical research findings is sometimes extremely difficult. It starts with phase 1 studies. So I did just a little bit about phase 1 studies. So traditionally in the area of clinical, we've always thought about phase 1 studies being the first introduction of a drug or a molecule into the patient or into the patient self, the human population, even before individuals that are healthy subjects, not even those who have disease or might have abnormal physiology, might not be the target for the molecule or the intervention. Of course, that's changed a lot. That's changed with cancer therapy, where individuals phase 1 clinical trials cancer are often in individuals who have the disease who may not have other therapeutic options. It's not the same gene therapy. And most recently, as we've seen with the transplantation of a porcine heart into an individual, it's not the same in things like xenotransplantation. And I do wonder if we can't say the same thing about microbiome-based research as well. If it will always be phase 1 research will always be in healthy individuals or in individuals with potential targets for the microbiome. So one of the unique things is it does require, as Daniel said, new thinking. And so it's just the novelty of the development of a compound or molecule or therapeutic, but also specifically around study design and also the ethics around study design. And just to step back, there's a particular series of ethical principles towards phase 1 trials and the protection of subjects, that is healthy, normal controls or volunteers, there's a different set of principles around phase 1 clinical trials in individuals with cancer for gene therapy. And I'll suggest that what we're starting to see now is also different ethical considerations design. So for example, Charles Weyer from the University of Western in London, Ontario, has led international groups on step wedge cluster randomized controlled trials, and how there are some different ethical thinking around that. Simply to say that it becomes a very and dirty complex, so I should say dirty and a real difficulty controlling many potential confounding factors or covariate features related to the disease where you want to do your investigations. And then layered on top of that, of course, substantial difficulties in a regulatory environment, a design environment, and an understanding of the ethics. So I'm going to pause my comments there, simply to say again, how impressed I was with the presentation that Daniel has made. B, how impressed I am with novel findings that arise out of the laboratory and the bench. And C, when things don't work out at the bedside, it's not always because it's not always our fault or that a compound doesn't work. Just sometimes the complexity that arises in the uncontrolled environment, that sometimes the bedside, the hospital, the clinical conditions. Very much.